So today we're going to talk about lipids, nucleic acids, and proteins. So first we're going to talk about lipids. So lipids are a class of molecules that do not include true polymers or monomers. They're generally small in size. They're often considered or not considered to be a macromolecule. Lipids are non-polar and hydrophilic. So hydro meaning water, philic meaning afraid of. They are afraid of water. And we can see this, right? If you've ever mixed oil, which is a lipid, and water together, they're going to separate. So there are different types of lipids. We have fats, phospholipids, and steroids. So fats are comprised of a glycol and a fatty acid. And a glycerol is classified as an alcohol. A fatty acid is a large chain of carbons with a carboxyl group at one end. So the three fatty acids join to glycol, and they can be classified as either a saturated fatty acid or an unsaturated fatty acid. Saturated fatty acids mean that they are saturated with hydrogen. There are no double bonds between the carbon and the carbon chain, meaning there are more hydrogens. And unsaturated means that there are one or more double bonds, meaning that there are less hydrogens in the mix. More double bonds mean more unsaturated, and more unsaturated means more liquid at room temperature. Bends and kinks prevent these fats to pack closely together. So you can see, right, like olive oil is an unsaturated fatty acid, so there is liquid at room temperature, right? So avocados, oils, nuts, things like that are unsaturated, while saturated fatty acids are going to be like cheese and butter. So phospholipids are a major component of cell membranes. They have two fatty acids attached to a glycerol and a phosphate. So we can see the phosphate up here at the top, and then we've got our glycerol, and then we've got two fatty acids. One is saturated and the other one is unsaturated. And they assemble as a bilayer in H2O, meaning that the hydrophobic, or afraid of water, tails are on the inside, and the hydrophilic, or they like water, heads are on the outside. The tails are hydrophobic and the heads are hydrophilic. So hydrophobic doesn't like water, hydrophilic does like water. So steroids is the last type of lipid we're going to talk about, and they have 4 fused rings. They're a unique group attached to a ring determined by the rings determine the type of steroid. So an example I've shown you is testosterone. Steroids are lipid molecules that are used to make hormones. Hormones are long-distance cell signaling molecules responsible for growth, development, metabolism, and homeostasis. Cholesterol is a great example of a steroid, and it stabilizes cell membranes to ensure fluidity. Too much cholesterol can build up in blood vessels, causing blockages. So you don't want too much cholesterol, but you want enough to stabilize your cell membrane so they don't fall apart and disappear. So now that we've talked about lipids, we're going to talk about nucleic acids. And nucleic acids are polymers made up of nucleotides that are the monomers. And the function is to store, transmit, and express hereditary information. There are 2 forms, right? We have deoxyribose nucleic acid, or DNA, and ribonucleic acid, or RNA. So nucleotides make polynucleotides form into amino acids. And nucleotides contain 3 parts. They have a nitrogen base, a 5-carbon sugar, or something called a pentose, and a phosphate group. In polynucleotides, each monomer has 1 phosphate group. So you can count the monomers by how many phosphates there are. So we can see it here, right? We've got a phosphate group, our 5-carbon sugar, and our nitrogenous base. And there are 5 nitrogenous bases. We have adenine, thymine, guanine, and uracil, as well as cytosine. And then our 5-carbon sugar is either going to be deoxyribose or ribose, depending on which nucleic acid we are making. So we have 2 pyrimidines and 2 purines, depending on which one we have. Pyrimidines are a 1-ring shape with 6 atoms. So these 3 are our pyrimidines, right? 1, 2, 3. So cytosine, thymine, and uracil. Thymine is only found in DNA, while uracil is only found in RNA. And then our purines are 1 ring with 6 atoms bound to 1 ring and 5 atoms to another. So the purines have 2 rings, while pyrimidines only have 1. We have adenine and guanine. I'm not going to have you know the difference between adenine and guanine or cytosine, thymine, and uracil, but I do need you to know that purines only have 1 ring pyrimidines only have 1 ring, while purines have 2 rings. The 5-carbon sugar base in DNA is the sugar dextrose. In RNA, it's just ribose, so deoxyribose or ribose, depending on which one we need. They differ in structure, therefore they differ in function. So we can see deoxyribose doesn't have that extra oxygen that ribose has. Like I said, not going to make you memorize these, but I want you to know that there is a difference, and because of that difference, they differ in structure, meaning they differ in function. Then our phosphate group is added to the 5-carbon sugar, which is attached to the base to form that nucleotide. So remember what I said, each phosphate can dictate a nucleic acid. So polynucleotides. So the phosphate group links with adjacent nucleotides to form a phosphodiester linkage. Not important, I just need you to know that they link together through the phosphate group, and they're directional, so they go in one direction. They go in a 5' to 3' direction. So we have a nitrase base, a sugar phosphate backbone, and the 5-phosphate end, and then we have a 3' hydroxyl end. And this will come up later, but I want you to understand and look at this now and see, and we'll come back and make connections later in different chapters. But it is very important that we have a 5-phosphate end and a 3' hydroxyl end. The sequence of the bases along the DNA or RNA is unique for each gene. So dictates the amino acid sequence, and also dictates the primary structure of a protein. So structure dictates function. So the structure of the DNA will dictate the mRNA, which will dictate the amino acid structure. That amino acid structure then dictates the protein. So if you think about it, our DNA dictates the protein shape and therefore the 3D structure. So DNA consists of 2 polynucleotides, and it forms a double helix. The strands are anti-parallel, so they are opposite of one another, and they are held together by hydrogen bonds between the base. So you can see them here, right? They are anti-parallel, they are the opposite of one another, and they run against or opposite. And the bases, so the nitrogenous bases form the rings of the ladder, right? So if we look at this like a ladder, these are our 5-carbon sugar and our phosphate, and right here are our rings. Anti-parallel, right? So this is our 5' end, this is our 3' end, And cytosine binds to guanine, and adenosine binds to thymine. And RNA is single-stranded polynucleotides. It varies in shape due to the base pairs with RNA. Adenine binds to uracil, and cytosine binds to guanine. All right, so now we're moving on to proteins. And proteins are formed starting with amino acids, and then we make a peptide bond, and then we make a polypeptide, and then our protein, which is our largest of all of them. So proteins are molecules consisting of polypeptides, and these are polymers of amino acids, and these are then formed or folded into 3D shapes. So if you think back to our protein folding lab, we eventually made a 3D shape of hemoglobin. They are comprised of carbon, hydrogen, oxygen, nitrogen, and sulfur. The shape determines the function. So the shape or the structure of the protein determines what that protein is going to do. So molecules, right, we've talked about amino acids are monomers. So molecules have an amino group and a carboxyl group. So our amino group is here, right? It's bound to a nitrogen, and our carboxyl group is a COOH. And they have 20 different amino acids. And the reason we get different amino acids is because of our variable or our side chain, or our different or a variable functional group, right? So we've got two examples here. We can see that amine group, that carboxyl group, and then that R group down here is different for these two amino acids. We have to remember the structure of the amino acid will dictate the function. So if the structure, right, if we add a carbon or a hydrogen or an oxygen or a sulfur, it's going to change the function of that amino acid. So you're each unique, making the structure different, making their function different. So each amino acid has a unique side chain, and the unique aspect of the amino acids are based on the side chain's physical and chemical properties. So side chains can be grouped as nonpolar, polar, charged or ionic. These side chain interactions will determine the shape and the function of the protein. Right? Do they like to be with water? Do they like to be without water? Do they like to be with each other? Do they not like to be with each other? It will all change how that protein is shaped, meaning that it will change the function. So, to form a peptide bond, the carboxyl group of one amino acid must be positioned next to the amino acid group or the amino group of another amino acid. They come together once again through that dehydration reaction to form a backbone where the side chains are out against the rest of the world. So we can see here, repetitive backbone, nitrogen, carbon, carbon, nitrogen, so on and so forth. And then we've got our side chains down here that make each amino acid different. Polypeptides, many amino acids linked together by a peptide bond through that dehydration synthesis reaction. Each polypeptide has a unique sequence of amino acids and is directional, so it is read in a single direction. And each end is chemically unique. So there is one free end of the amino group called the N-terminus, and then one free end of the carboxyl group called the C-terminus. The sequence of the amino acids determine the shape. Structure dictates the function. Remember, shape determines function. And when polypeptides twist and fold, because of R group interactions, it forms a protein. So take a second and think about how is the unique structure of amino acids determined for a polypeptide sequence. You might not know this answer, and that is okay, but I want you to think about it and then share with a partner what you think the answer is. Genes, right? We talked about it with nucleic acids. So the function of proteins include Antibodies, so it helps to protect our body from diseases. Enzymes that carry out chemical reactions or assist in creating new molecules. Messengers that send signals like hormones. And then structural, they provide structural support for our bodies as well as transport and storage, so they can bind and carry small atoms and molecules throughout the body if our body needs it. So we talked about structures and how proteins fold, so let's quickly talk about the 4 levels of protein folding. So we have primary, which is just a linear chain of amino acids, and it's determined via our genes. The direct dictates the secondary and tertiary forms based on those side chain interactions. And then we have secondary, which are coils and folds due to hydrogen bonds within the polypeptide backbone. The beta sheet and hydrogen bonds between polypeptide chains are laying side by side, right? So we have beta pleated sheets, so they pleat together. And then we have an alpha helix or spiral, and we have a hydrogen bond between every 4th amino acid. Right, so beta pleats, alpha helix, so folds and spirals. So we have tertiary structures, which is 3D foldings due to interactions between the side chains of the amino acids, and it's reinforced by hydrophilic interactions. So these, we have beta sheets and alpha helixes start folding in on each other. And then we have quaternary, which is associations of 2 or more polypeptides. They are found in only some proteins. All 4 levels of protein structures determine the function of the protein. Once again, structure dictates function. So now that we have looked at all of the different macromolecules, let's put it together in a chart. So we have carbohydrates. They have carbon, hydrogen, and oxygen, and they are a monosaccharide and a polysaccharide. Then we have proteins, which are carbon, hydrogen, oxygen, nitrogen, and sulfur. Their monomers are amino acids, and their polymers are polysaccharides. Then we have lipids, who have carbon, hydrogen, oxygen, phosphorus for phospholipids. And then their monomers or subunits are glycerol and fatty acids, and they don't contain any true polymer. And then finally are our nucleic acids, and they contain hydrogen, oxygen, nitrogen, and phosphorus. Their monomers are nucleotides, and their polymers are DNA and RNA. That is the end of my lecture. If you guys have any questions, please don't hesitate to reach out. I will see you later. Bye.